We're talking about drug therapy, a mental health necessity. Forget about your worries and your strife. Forget your CBT, don't mention psychotherapy. Biology says pills sort out your life. Hello and welcome to drug therapy, which is the preferred therapy of the biological approach. I'm going to go fairly swiftly through this, as it's my fourth attempt at recording it. Don't ask. Okay, so what do you really need to understand, big picture-wise, as far as this goes? You need to understand why a biological psychologist would actually choose to use drugs to help people with problems. And you also need to know how those drugs work, and to be able to evaluate whether whether it's the right thing to do, whether it's a good thing to do. So they're the key things, big picture-wise. Now let's have a look at learning focus, things that you do need to know. You need to know that how the biological assumptions apply to drug therapy. So you need to be able to make the link between using a therapy, using a particular type of drugs, and how that's connected to the assumptions of the approach. You need to know the main components of the therapy, which means you need to know how they actually work. So I'm going to take you through how antidepressants work, and to show stage by stage how they work, and how antipsychotic drugs, so drugs for schizophrenia, how they work. Okay, so you need to know how both those types of drugs work. The book's also got anti-anxiety stuff, but I'm only going to cover two in this talk. Then you need to be able to evaluate the effectiveness of it. So you need to be able to quote research showing that whether indeed drug therapy is effective for depression, schizophrenia and stuff like that. Okay, and then you need to, again, like I said in the big picture point, you need to be able to evaluate whether giving people drugs is the right way to deal with a particular issue. Okay, so how do the biological assumptions apply to drug therapy? So what is that link? So as we know, the biological approach assumes that all behaviours have got a physical cause. And if that is true, if that's the assumption, then we would think that behaviours like, or disorders like schizophrenia, depression, anxiety, they've got a physical cause, and in the same way that physical illnesses do. So if they've got a physical cause, in the same way that we give people drugs to help them with physical illnesses, drugs should also be able to help people with psychological illnesses. Another of the assumptions of the biological approach is that neurotransmitters affects behaviour. So the level of neurotransmitters affects behaviour. So you would expect drugs to be involved in altering levels of neurotransmitters, and we'll see that that is true. Also, the assumption about localization of function. Different areas of the brain are responsible for different aspects of behaviour. So those areas could be targeted by the biological approach as well. So there are a lot of very strong links between the biological approach, assumptions, and its use of drug therapy. Okay, and you need to be really clear about those are. So you might want to pause it for a moment and just check that with yourself. Just ask yourself a question, and then maybe have another look at this. Right, next one. So this is just a little bit of revision about how nerve cells work, of course. So we have the green blobs in the middle. That's the nerve cell body. That's the main part, or the nucleus of the nerve cell. The long spidery bits are called axons, and then the gaps in between your nerve cells are called synapses. Okay, so nerve impulses pass along one synapse, and they come to the next nerve cell, but they have to cross a gap called a synapse. Or a synapse, I don't mind how you say it. Okay, so the electric signal travels along the length of the axon. When they get to the end of the axon, also known as the axon terminal, chemicals take, or neurotransmitters as we call them, chemicals take the impulse over to the next neuron and stimulate that electrically as well. But oh crikey, that's big writing, isn't it? That must be an important point then, eh? So the important point is that drugs act generally by interfering with whatever's happening at the synapse. Okay, and we'll see that in the two examples. Okay, so let's just look at how a nerve cell works again. And a nerve impulse travels along from one neuron to the next. So the nerve impulse is travelling down this way from the top, and it comes to this thing here, which is the axon terminal. And in the axon terminal, there are loads of vesicles, little round things containing neurotransmitters. So the red arrow things are neurotransmitters. Okay, so the electrical impulses forces the vesicles to open and split all their neurotransmitters into the synapse. So these neurotransmitters are looking for somewhere to go, and they're looking for receptor cells, which are these ones down here. And look what happens. This is remarkable, and this is happening all the time. This is happening as I'm speaking to you millions of times. It's happening to you as you're listening. Look what happens to this. The neurotransmitters come along, and they hit the receptor cells. And this little thing here means that they're activating them. So the chemical along here, it activates these receptors, which means that there's now an electrical charge which travels along the length of this neuron, carrying on going down, if you can see my little cursor thing. Okay. And then, interestingly, we want to know what happens. Well, enzymes are released to break down these neurotransmitters. We don't want the neurotransmitters in the synapse all the time, so they break them down. Any that aren't broken down by enzymes are taken up by some kind of what they call a transporter mechanism back into the presynaptic neuron, and they'll be recycled. Look, they're put back into vesicles and they'll be used again. Okay, so that's how it would work normally. Okay, that's how it works all the time for all of us. Now, what we have to figure out is what's happening if someone's suffering with depression or schizophrenia or whatever, right? And what we know about someone with depression is that it's generally associated with low levels of serotonin. Okay, so what we want to do is that we want to increase the amount of serotonin that's actually present in the synapse. 4 ways that we could do this. Really, the bottom 2 are the ones that we're most interested in, and really, I'm going to say most about the final one actually. So we could have drugs that stimulate the production of more neurotransmitter. We could introduce a chemical that acts like the neurotransmitter, so it could be like a fake neurotransmitter that somehow boosts the level of what seems like serotonin. The slightly older type of drug that's not used so much now is what we call a monoamine oxidase inhibitor. Right, so monoamine oxidase is an enzyme. Okay, so if the enzyme breaks down serotonin. So if we could actually figure out some kind of way of stopping the enzyme, reducing the amount of enzyme that's around, then we'd have more serotonin in the synapse. So that's one way of doing it. The more modern way of doing it is to stop the mechanism that takes the serotonin back into the presynaptic membrane. Okay, so that's the most modern way of doing it, and those drugs are known as SSRIs, selective serotonin reuptake inhibitors. Selective serotonin reuptake inhibitors. So they're selective because they choose serotonin amongst all the other neurotransmitters that are flying about. They choose serotonin. Reuptake inhibitors. So they stop that mechanism whereby the serotonin is taken back into the presynaptic neuron. Okay, so here we go. Here's how it works. Look, so here comes some serotonin into the synapse. There they go. Binds to the receptors, stimulates the cells, as we've seen before. Now, what the SRIs do is that they prevent the serotonin being recycled. So it kind of slows down that whole recycling process, which keeps it in the synapse for longer, ready for the next impulse. Okay, so schizophrenia, on the other hand, is associated with too much dopamine. That's that. As you'll find out in year 13, there's more to it than this. But for now, this is the picture that we have. Okay, so what we would want to do is somehow decrease synaptic neurotransmitter activity. So either by increasing the rate of neurotransmitter breakdown, so increasing the amount of enzymes that will break down dopamine, or the more common thing that we do, and the two types of drugs we're going to look at are do this, which is in actual fact blocking off the receptors. So let's have a quick look at that. So we've got two types of drugs that block receptors. Okay, so there's either conventional antipsychotics. These were the first ones, sometimes called first generational antipsychotics, also called typical antipsychotics, because they were the most common at one stage. And a good example is chlorpromazine. So what they do is they bind to the dopamine receptor without activating them. And that's really important, that they don't activate the dopamine receptors, because what we're talking about there is in schizophrenia, people often talk about somehow being overloaded with input and being just really overstimulated. So if we can actually stop people's neurons being too stimulated by blocking off receptors, then that's what they're doing. And both types of antipsychotic do that. It's called, by the way, they're called antipsychotic because schizophrenia is a psychotic disease, which means that you lose contact with reality. So an antipsychotic drug is designed to put you back in touch with reality. Okay. So what they do is that they both bind onto dopamine receptors. DA stands for dopamine in this, without activating them. But just a slight difference. So look. So we have this is a chlorpromazine drug, and look, it gets really stuck into the receptor molecule, which means therefore that it takes a long time for it to come out again. So it's got a slow release. This one, on the other hand, this would be clozapine, an atypical antipsychotic. Comes in, it blocks the receptor, stops it being stimulated by dopamine, but then it just slips out again a lot more easily. Okay. And this is what it looks like in here. If you think of these triangles as antipsychotic drugs, it's blocked all these receptors, so the dopamine in this case can only stimulate a couple of cells, which means that the person won't feel so overstimulated as they would have done if there was no drugs in the system. Okay, I think I'm going to stop there for a moment. I'm going to go back. Okay, I'm going to stop this one there, and I want you to now pause the video and just from memory, jot down what you can remember in terms of those objectives. So it's about understanding. Oops, sorry. So it's understanding how the antipsychotics and antidepressants work. What are the links between antipsychotic and antidepressant drugs to the assumptions of the approach? So see if you can remember those two. I'm going to save this so that I don't lose this video as well, and then in part 2 we'll look at the effectiveness of drug therapy. Okay, so thank you for listening. Oops, done it again.